soon, if you're ready.
Very unfortunate. Thank you for still being present. To all of you who are still present. Again, I kept the original title, Genotypic Data Analysis, and then subtitle, Stack the Odds in Your Favor. Because like I see it, genotypic analysis mainly serves to increase your probability of success, so getting a higher rate of genetic progress. We've been over this, I've been over this, other speakers have been over this. So I guess I can go fast over QTL, and I can go fast over GWAS, and I can immediately start talking the dirt about GWAS. So, remember, genome-wide association studies, the idea is finding one, but preferably multiple SNPs or markers that are associated with the trait of interest. Now, this undoubtedly works brilliant if you're working with Mendelian traits and you find a marker that is associated or maybe even on the resistance gene. But in my humble experience, GWAS has not been very predictive for most quantitative traits, if not all quantitative traits. And I'm not alone in this observation. If you look at the cartoons below, where people have been jokingly claiming that a large part of the heritability of the genetic variance is unexplained by the markers that you find in a GWAS study. And if you noticed that in most GWAS studies that are being presented or are published, you'll notice for quantitative trait, the amounts of variation they explain are usually very, very small. That doesn't mean that the association isn't real. I'm not claiming that it doesn't work. I'm just claiming that the percentage of genetic variance, the heritability that you're predicting, between 5 or 10%, and 10 would already be a very good GWAS study, is far too low to be using in a breeding context. Don't throw things at me. So, reversely, many of the associated variants are not causal, meaning they're associated, meaning that they might even break linkage, although that is more of a problem in QTL studies than admittedly in GWAS studies. But anyhow, there's always missing heritability. There's always a very large chunk of genetic variation unexplained, and it is very unlikely that GWAS will be the solution to that. Now remember my introduction, if you remember this morning, on quantitative genetics, where I claimed that the vast majority of economically important traits in plant and livestock species show a continuous distribution, aka is a complex trait. Now, there are basically two major theories, which are practically the same, that explain genetic variation for quantitative traits. First, you have the infinitesimal model, which is used in calculation of blobs and so on, which claims that there is an infinite number of unlinked additive loci, which each have an infinitesimally small effect. Basically, saying it is impossible to ever find and explain a quantitative trait, because the number of loci is infinite, and each locus has such a small contribution that you'll never pick it up. Now, why is this kind of theory put out there? Because that allows for a lot of simplification in in quantitative theory, in equations, and so on. So the big boys like Fisher and Henderson, who really, like Fisher is the inventor of the analysis of variance, Henderson is the inventor of the mixed model equation. So these are not the small fish here. These have always used this infinitesimal model in all their derivations. Now, the reality is probably closer to the truth is the finite loci model, which basically says that the number of loci is not infinite, but just really big. And really big is very relative to the trait you're looking at. Now, the picture on the right is not something special. I just typed in Google gene network and then took the most complicated looking image and put it in my presentation. But the goal is to show here what you're up against. if you're doing a GWAS study, you're trying to, first of all, you're trying to find each an effect. One of these genes might have an influence, but might be corrected by others. All these things are interacting with one another. With one another. So it's highly unlikely that you will be able to unravel each of these genes and their functions in traits like yield, earliness, and so on. That doesn't mean that you have to start desperately crying as a breeder, but showing you why GWAS actually is probably not the optimal solution if you're looking at quantitative traits. Now, so this is the first one is basically saying the same. Imagine that you have QTLs, that you found some associations. If you try to stack them, in general, for complex traits, this won't bring you far. There are exceptions. I know there are success stories, but they're not big and they're not the standard. In generally, you'll find some QTLs, and if you try to stack them, your contribution to the total breeding value, your rate of genetic progress will remain more or less constant. I know this is a very dim view on GWAS studies, so sorry for all the people who are now actively doing that. Now, one of the key points, so now it's turning into a more positive story from bullet point number 2. The emphasis, if you're a breeder or working in a breeding program, is predicting the genetic merit of individuals or their parental capabilities, and not really discovering the causal genes. From an academic point of view, finding the causal genes is of course brilliant if you succeed, but as a breeder, and especially what I would call old school breeders, have never been looking for causal genes, why this one variety performs better than the other. So, prediction of breeding values does not necessarily require the identification of the underlying genes. So what I'm proposing here is that you let go of the Mendelian thought that you can find the causative genes for your trait, and more work like breeders, traditional breeders are working, trying to find the guys with the best breeding value with whatever method is available to you. And the title of the slide is genomic selection. So that is the idea of genomic selection, not trying to find causal genes, not trying to explain the trait, but trying to predict the trait. And the two seem so closely related that most people have hard to distinguish. But the entire idea, the entire mechanism is different. So this is, in case the cartoon isn't clear, this says phase 2 interpretation of the genome, and it's a puzzle of 3 billion pieces that they're trying to find. So this is, again, a kind of a picture showing you what you're up against if you're trying to find the causal genes. And that is not what genomic selection is for. So this one you've already seen. We try to trace all the QTLs in the genome using a large number of markers, and possibly all markers can have some effect. First introduced, at least the term is coined in 2001 by Maenhout, and is already routinely applied in many plant and animal species. This one I think you've already seen as well in the presentation of Isabel. So the idea, I'll be very fast, you have a reference population of pigs this time, you genotype them and you phenotype them, you create a kind of a prediction equation, we'll talk about that later. Then you have a selection population, all candidates which have only been genotyped, and using the prediction equation, you can select those with the genomic breeding values, superior genomic breeding values. Notice again that I keep using the word breeding value. I'm not using the genetic value or per se value. I'm not looking for the top individuals, the top performers. I'm looking for the best parents. So the principle stays. So I'm very consistent over my 3 presentations. This is exactly the same, but shown in a kind of a more mathematical way. Your training population is basically nothing more than a vector of yield scores, for example, and a matrix of markers. So the line is an individual, and then each column, marker 1, marker 2 to marker P, is the score for a certain, let's say, SNP. So this is your input data to train a model. These guys, you don't have the yield column, you make predictions, and you select the superior individuals. So again, looking at genomic selection or genomic prediction as a kind of recipe, what is needed? Well, first, you need a training and a reference population. So for each individual of that training population, you need a single phenotypic response. The word single is important. Most, if not all, genomic selection methods, not all, most, assume you have a single phenotypic response value for every individual. Most plant breeders have multiple phenotypic values. You have multiple trial results, multiple estimates, and multiple years. So most methods assume that you have one single response and a complete set of molecular marker scores for every individual. Again, the word complete is important. As Tom showed, most genotyping methods will have missing data at certain levels, and it differs among technologies, but it's very rare that you will obtain a complete set of markers, scores. Then you need a genomic prediction model, a kind of mathematical statistical approach to learn phenotypic response. So here I am in contradiction. To learn breeding values from marker scores, and then a selection population where you will apply. Now, this recipe will return. Training population-wise, there are two main strategies that I see used in practice. So this is actually in breeding companies. How do you get this training data for making these genomic prediction models? The most popular strategy to start your adventure in genomic prediction is to make use of historical data. Now, if you remember my talk on phenotypic data analysis, I said that data archaeology is one of the more challenging things to do, because historic data is usually incomplete, unbalanced, and problematic in several ways. Usually not always. So basically they will use phenotypic data that is available from their traditional breeding program and genotype individuals that are still alive for genotyping. Strategy number 2 is what companies do that routinely use genomic prediction. That is where it is the standard de facto way of selecting. They will generate data from dedicated training panels. So at that point, your breeding company, a large proportion of your phenotyping efforts are not for finding trial candidates or finding superior individuals or superior parents. Your phenotyping efforts are made solely for training genomic prediction models. Now, there's a big step to make for a breeding company or an institute to go from 1 to 2. Because in 2, you're actually sacrificing trial space to train your genomic prediction models. Nevertheless, it is the most optimal way if you want to have high efficiency genomic prediction like you read it in the books and in the papers. It's very hard to convince people to go from 1 to 2, to go to 2 directly, to skip 1, because it means you really jump in, invest money, and do everything without ever knowing what the returns will be. Now, using historic data, advantages should be clear. You don't need to invest in any resources for phenotyping. Disadvantages, there are a few. Like I've mentioned, phenotypic data is generally severely unbalanced. You need advanced aggregation methods. That's basically these multi-environment trial analysis I talked about in the previous talk. Aggregating over years, location, trial, and this one is important. There's a strong selection bias. So normally, the bullet point below that says that you generally have an over and under representation of parts of the genetic space. So if you look at your breeding pool as a kind of a space where every individual has a certain position compared to one another, where everybody's, well, more or less related, if you use historic data, you actually train the model exactly on that distribution of the genetic space. But imagine afterwards, it has been brought up by a question after Isabel's talk, I think it was, that what if you want to use your genomic prediction model to learn something from individuals that are outside of this space? Well, this historic data won't help you there. So at that point, your genomic prediction model is confined within the genetic space of your historic data, within that selection of individuals. So size and genetic range of the training population is predetermined. And one very important is that usually there's a strong selection bias, meaning there's a high overrepresentation of good individuals versus bad individuals. And that is the biggest problem, actually. I should have put that one in bold. Because if you want to have a fair genomic prediction model, you need an equal representation of good and bad. If you want to have a prediction model to separate good from bad individuals, you have to provide it a proportional amount of individuals for good and bad. Now, it's very, very hard to convince breeders to put bad varieties in trials, not kill them, but score them, keep phenotyping them until the end, just like the good guys, and not in a very small amount, but in equal proportions. So historic data is never like that. Historic data is strongly skewed towards good individuals, at least if you've been selecting right. So at that point, reduced prediction accuracy is to be expected. How much? It very much depends on the severity of the skewness and other things. But basically, if you see the use of historic data as a kind of a shortcut to get a bit of a feel what genomic prediction could do for your breeding program, you're actually limiting the potential of the technology. You're actually underperforming because it can do way better if you're willing to use dedicated training panels. Oop. So dedicated training panels, you can tune the size of the training population and the relatedness to the breeding population. So it's a bit of an art now to construct training population. There's no standard way to do this, meaning there are several studies. I think it started out in 2012. Until now, there are still publications on the topic on how do you construct an optimal training population for a certain breeding pool. And one of the things, and I think Isabel also mentioned it, is that the distance to what I call the breeding population, to the population you're willing to predict, that you want to predict on, is quite crucial. So I often see genomic prediction models where they say we're going to use all the data that we have and then use the model to explore different parts of the genetic space that we've never visited ourselves. Well, that's just a contradiction. That will never work. If you want to make predictions on an uncharted territory of your genetic space, you'll actually have to collect phenotypes in that part. And it also generally means that your what I'll call the resolution of your genetic space. I'll use these wide things to mean that this is a wide genetic space. So you have lots of individual, and guys that are here are completely unrelated to guys that are over here. If you want to train a model that can make predictions in such a wide area of the space, you'll need a huge amount of training examples, depending on species, of course, and depending on linkage decay and so on. But generally, if you want to have a working over a very wide space, you need a big amount of training examples. If you're willing to say, no, I'm just going to focus on a small part of the genetic space, like this is where I'm working in, then you can reduce the number of training examples. And that is what you do when I say tuning the population and the relatedness to the breeding population. That is what you decide. So you have, in companies, they often have different training panels. You have the broad spectrum training panels that do exactly that. They make predictions over the entire genetic space. Each prediction is not that good, so it's a low reliability prediction, but it gives you an idea where you are in space. And you have these high precision genomic prediction models that focus on parts of the genetic space that are much more narrow and needs less training examples. And that is what dedicated training panels can do. Important, getting a balanced representation of good and bad training examples, and getting quality of phenotypic data. So number of environment, replications, all the things I said in the previous talk. obvious disadvantage, you need additional resources. And you need a different mindset. It's like I said, and I said it jokingly, but it's very hard for a traditional breeder to phenotype bad performing plants. Nevertheless, that is important. Finding a scale that allows you to balance, to make observations from good to bad that are very representative, because that will be the scale where you get your genomic predictions in. So I think Isabel also mentioned it, the relationship between training and target population is the key to accurate genomic predictions. If they're closely related, only small training population is needed, but your genomic prediction model will only be useful within that genetic range. If they are completely unrelated, large training population is needed, and your genomic prediction model is widely applicable. Now, I'm not sure if this last sentence gives a lot of hope, but in theory, the reliability of your genomic predictions will approach 100% if your training population goes to infinity. Now, that doesn't mean much, but the idea is basically what I see in plant breeders. If you compare it to Holstein, so the cattle breeders, they've been using genomic prediction, I think, were very early adopters from the early 2000s. And they're working on one species, cow, and then one variety, namely Holstein, which has a very small effective population size. And they have training sets that encompass hundreds of thousands of animals. Not data records, not milk production, but actual animals. So they have these huge data sets, and they struggle to get, well, they don't struggle, but they get decent reliabilities on their genomic prediction models over all Holsteins they encounter. Now, I've never seen in plant breeding a data set that contains 100,000 individuals. Add to that you're not working in one particular part of the genetic space like the Holstein is in the entire cattle space, genetic space. No, usually, and this is especially true, I guess, for forage breeders, you're working in a huge genetic space. So the dream of making a genomic prediction model that will be valid for your species is possible, but it will take you a huge but enormous training population to learn that. So it's practically impossible. So you have to learn how to use that. You have to give up the idea of finding the prediction model for your species. You have to find the prediction model for your breeding pool or that particular part of the breeding pool. If you're going for training panel, like I've said, there are many ways. There is currently no de facto standard way of creating a training panel for genomic prediction. But all these methods have more or less one thing in common. The main goal is to prevent the selection of closely related individuals in the training population. Because if you have one entire family in there, first of all, they don't contribute much on the LD level because they probably all have very similar LD blocks. So they don't contribute very much, but on the other hand, they are dominating the training panel. Because like I said, the training panel, the relatedness with your selection population is very important. So if that family that you have put in there is very unrelated to your selection population, your genomic prediction model is worthless. So you try to find individuals that are as unrelated as possible, but still representable for your genetic space. And then there are different ways using, and these are all linear mixed model-based methods. So the PEV is the prediction error variance, or the generalized coefficient of determination. The CD, these are basically just reformulations. The CD is a reformulation of the prediction error variance. These are all things that result from a mixed model analysis of your phenotypic or genotypic data. So you see that all of these methods, starting from what I said, the phenotypic analysis, towards genomic selection, towards training set construction, they all rely on the linear mixed model framework. So you see that it comes back in almost any of my presentation. An optimal method, turns out, currently depends very much on the species, the breeding pool, the marker type, the marker density they're using. So I cannot really say this is the way forward for training panel construction, but there are already several methods out there to do that. This is again a bit of a showcase. I didn't put the reference in here, which is bad. This is not one of my studies. But this is a simulation study of what happens if the distance between your training population and your selection population gets bigger. So it's very similar to what Isabel showed. With the extra detail that is a simulation study, because what you're looking at is the FST, so there's a measure of distances between populations, and you start out here with your training population, and you start selecting, and your next population that you arrive will have a genetically slightly different from the original population. And you could keep on doing that. You don't update your genomic prediction model. So you keep on just using the same genomic prediction model. And we assumed here, contrary to my claims about quantitative traits, that there are only 10 QTLs determining the entire trait. Now what you see is that there is an immediate drop almost as soon as the populations diverge in the accuracy of your genomic prediction model. Most likely because your LD gets broken quite rapidly, because you only have 10 causal variants, so each of them are quite important. As soon as you lose one of those, prediction accuracy drops, and you're very rapidly, with an FST of 0.1, you're at a prediction accuracy of 0. Now, it's not that bad in reality, because if we're looking at 1,000 causal variants, which is still not a huge amount, we see that it's not that extreme, but it's still pretty bad. First of all, it's more difficult to start with. The more genetic variants that you have, the more difficult it is to get a good prediction from the start. So it's like a lower heritability. But then the drop is not that sudden, but you see we're already at 0.1, we're at 0.2. So it's not 0, but it's not really a prediction model. So prediction beyond the training set is very hard. That is basically the message. It depends a bit on the architecture of your quantitative traits, how much QTLs are involved, but in nearly all cases, even if there's a large number of QTLs, the drop will be quite considerable after a few generations of selection. So this was kind of in part one. What do we need for genomic selection? Remember the recipe. I've put it in green. We need a single phenotypic response value, preferably measured as a continuous variable, because like I've mentioned, scales and things like that are more difficult to do. This one you've already seen. So the machinery that I've introduced in previous talk about multi-environment trial analysis would be exactly what you need here to get a single phenotypic response. It would be then a blob, for example, an estimate of the breeding value of an individual. But anyhow, you would need this machinery to aggregate all the phenotypic data that you have available. And that's why when I said that when you're a breeder and you're jumping from, we're just using the standard method of crossing the best and hoping for the best, and jumping to genomic prediction doesn't make much use as if you don't master this machinery. You need these advanced phenotypic analysis methods to get good phenotypic estimates to train your genomic prediction model. So they're all related to one another. Second one, you need a complete set of molecular marker scores. Now, seems easy, right? Now, unfortunately, like I've mentioned, marker scores generally have extensive missing observation. Extensive depends on the technology that you're using. Tom can tell you a lot more. They often also contain errors and inconsistencies, and more problematic, they are often non-informative, meaning a lot of markers will be on, if you have a certain training set and you have like a SNP chip of 300,000 markers, a certain percentage of that, depending on your breeding pool, will be completely fixed. They will all have the exact same score. That means they are non-informative, non-polymorphic. It means that is completely wasted. If there's no variation, you cannot use this data. Less obvious to see is redundant information. Imagine that you have this big SNP chip, and everywhere where on marker 1 there's an A, there's on a second marker there is a G. and vice versa. Everywhere there's a T, there would be on the second marker, there would be a C. Those two markers would contain the exact amount of information. So dropping one would give you exact the same result. Now that is redundant information, meaning correlated with other markers. So a third problem, well, that's the least of your concern, is that depending on the technology, marker scores are often coded as allele pairs, or if you're working in polyploids with multiple alleles, and you have to transform them into numeric data. But that is, in fact, not a real issue. Now, there is an entire field that has exploded since the 2001, the use of genomic prediction, which is called genotype imputation. I'm not sure if you're ever used or encountered the term, but the official definition is the statistical inference of unobserved genotypes. Basically, you have a matrix with genotypic scores. If you want to use that matrix to train a genomic prediction model, you have to fill in the blanks. You have to fill in the missing scores. Missing data means no training model, no prediction model. So what genotype imputation as a term means that you will have to find an intelligent way to fill in the missing data. Now, there are various ways and various reasons why you could do this. For example, this is I think, very common case. You have a small percentage of genotypes that have not been called. For example, less than 5%. So these are your individuals. Question marks means that you don't have score. Imputation in general, we'll talk about the different ways of doing imputation, will just complete the missing genotypic data with what is a logical solution depending on the method and the person that you're asking. So there are various ways to do imputation, but this is just showing that just to fill in a few missing data, that works pretty well. Another use of imputation is when you have two genotyping platforms. It's often the case when you go from one platform to another, as new technologies arise and you don't want to throw away your old data. You have overlapping, so some genotypes, there are only a few SNPs that are common. So these are all platform A scores, and these SNPs are not in platform A, and this SNP is not in platform B. But you still need overlap. If the two platforms give you completely different SNPs, then of course imputation does nothing. But this is a second use of imputation. And third one is very important, or was very important, I'm not sure, to make genomic prediction economically feasible. So this was actually the reason why genomic prediction was invented mostly. Imagine that you have these high-density SNP chips. There's a certain cost if you have to do this for all the material that you're screening, and you have to genotype them at such high density, there's a certain cost towards that. Now, the idea is here to use individuals and genotype them at very low density. Well, very low, that is something that you have to find out. But at lower density, and then try to impute from low density to high density. So basically, this is a way to do genomic prediction cheap. Because one of the things that you often hear when you talk to companies, they say, yeah, but genomic prediction is not feasible for my crop because the genotyping is too expensive to have these high density. But that is the case for most crops. Even for the big field crops, they will genotype at lower density and use imputation to go to high density for their screening material, for the early screening. You reduce the accuracy of your genomic predictions, but cost-wise, it is far more efficient than genotyping everything at high density. Last use, but this is really something ongoing, is the GBS kind of thing approach, where you have low coverage sequence data. So there's a bit of the same thing. You will use GBS, but use low coverage data and just try to fill in the blanks with imputation methods. But these methods to my opinion, are currently not very mature. So there's not a lot available for this scenario. But again, the idea is the same, using low coverage sequence data to decrease the costs of genotyping. Now, how do you do this imputation? I'll classify them in 4 different categories. First one, naive, meaning easy. Very popular, just use the marker mean. Imagine a column in the matrix and some parts are missing. You just take the average of the column and you fill that in the blanks. So that would be actually an imputation that is related to the allele frequency of that particular marker. There is even a more easier way that you fill in if you're working with inbred lines, homozygous individuals. Then you either, there's 1 or there's 0, for example. You have the reference allele or you do not. And if you don't know, you put 0.5. I hope you feel that these are not very advanced methods, right? Therefore, the naive imputation. Second category, basic statistical approaches, meaning approaches that have been existing long before genotyping and imputation, before those terms were even coined. For example, k-nearest neighbor clustering. In principle, very easy. You calculate a similarity matrix between all of your individuals. So this is the genotype scores from one individual I, this is an individual J, and you calculate the similarity. There are various ways, I think Isabel mentioned some. You calculate the similarity between individual I and J by, for example, multiplying their vectors of scores. You get some form of similarity measure. You put all of these similarity measures in a matrix. So this is the similarity between individual 2 and individual 1. This is 3 versus 1, and so you get the idea. So if you then have individuals with missing scores, you will take the major allele of the k nearest neighbor of that individual. So basically, you look at a hole in the matrix, like here you will find the guys that are closest to him genetically, using all the other markers that you have available, and then say what is the majority allele that occurs in those 3 other guys, where 3 is my k, it could be 5, could be 10. So this is also allele frequency based, but takes into account that related individuals will most likely share the same allele. Of course, the message before was you should train on unrelated individuals, so a bit of a contradiction there. Population-based approaches, I guess, are the most popular today, commonly used in human studies, so that's a bit the standard approach. And they use short-range linkage disequilibrium information in a population. So these will actually need the location of the markers on the chromosome to know which ones are close, calculating correlations. And then there are various methods that differ considerably. Most of them you use Bayesian and Markov models, hidden Markov models. They differ quite wildly in accuracy and majorly in computational requirements. The biggest player in this is Beagle. If you should Google it, it's one of the best in accuracy, getting the good imputations, but it's beastly slow. It takes like days to do the imputation on matrices. And these are like variants of the approach, but are a lot faster. So there's a lot of work going on. You see that these papers are not recent, right? So imputation has really started off approximately at the same time that genomic prediction was starting off, because the problem was immediately clear that if there are holes in the matrix, we cannot continue, and simply putting in the mean or something is just not working out. Family-based approaches, again, a bit of a contradiction if you're working with unrelated individuals, but these will then work with long-range LD, so real linkage. Again, different methods. A lot of benchmark studies going on, not clear who is the winner. You know the drill. Very much dependent on which breeding pool you're looking at, which marker technology, and so on. But just to give you an idea of the power, So these are the kind of studies that you would generally do as a breeding or as a breeder or as a breeding company to know and how the game is called how low can you go, meaning if we reduce markers, how much prediction accuracy are we losing? So this is a study from John Hickey in 2012 and Mays. And the idea is that we start out with a, well, for those days, high-density SNP chip of 35,000 SNPs. And we have a panel of 1,163 inbred lines. And I imagine a kind of a phenotypic response. So we gradually mask, well, we, or John, gradually masks genotypes, starting out first with just randomly masking 5% of the markers, and then going on to masking 50% of the markers, 75%. So you end up with only 25% of the markers being used, and you impute the rest. So it's not that you're working with a reduced set of markers. Each time you mask, you remove 50% of the markers, and impute all the missing ones with, in this case, this was John Hickey's own method, alpha impute. Now, if you look at the prediction accuracy, the correlation, this means between the true breeding value and the predicted breeding value, you see that you remove 50% of the markers, so ending up here with like 17,000 something markers, and you get overall quite the same prediction accuracy. Remove again half of the markers, ending up with only 25% markers. So on the horizontal scale is the percentage of markers that are masked, removed. And we see that it starts to drop a little bit. And this goes on, and then this here, the ones in red are a kind of the breaking point, where you see prediction accuracy actually suffering. We go all of a sudden to 0.8 and down. So the distribution are the different iterations of sampling, because you can remove 50% of the markers in very many ways. So he sampled the procedure several times to get an idea. But you see that it starts to break down as long as you go to 94 to 97% of the markers masked. But that's a huge amount of markers that you can remove before prediction accuracy starts to suffer. So that is the power of imputation. It seems a bit contradictory, because often people think, then why do imputation? Just do it on the reduced set of markers. Don't use the 35,000. Just use, let's say, if you're going to use the half of them, just use 17,000 markers. Well, it turns out that imputation and upgrading to high density gives better results than just using the reduced set of markers, which is a bit difficult to understand, because they both rely on linkage disequilibrium, both genomic prediction and imputation. So I'm going to assume here now that using multi-environment trial phenotypic analysis, that we have a single phenotypic response for every individual in our training population, and by using imputation that we have a full set of marker scores. These are the key components. The next thing that we need is a genomic prediction model, so a statistical model to learn phenotypic response from marker scores. Again, there's a plethora of methods. Don't worry, I won't go into details of any of them. But there is a bit of a mismatch. A lot of scientific literature about genomic prediction is about genomic prediction methods, where an inventor of a certain method A claims that it's better than method B and better than method C, then inventor of method C claims that the inverse, and so on and so on. So, like, most of the genomic prediction literature is dominated by a comparison of different methods. Most, if not all of them, are very clever, are very good methods, but it kind of gives the idea that the difficulty of genomic prediction is in finding a good prediction method. While in my experience, most of these methods have very similar results. And the big effort is actually put in phenotypic data analysis, so previous talk, and getting good complete marker scores will be vastly more important than choosing the appropriate prediction model. Now, again, similar I'll not go into details, but most methods rely on some form of linear model, some form of linear mixed model if necessary. That depends on the method. Now, question that arises, maybe I should give a little bit of intro first. In most cases, you'll have a bigger number of markers. So in this case, I think, no, in this formulation you can't see it, but imagine that you have this SNP chip with 35,000 markers. Those are 35,000 variables for which you're trying to get an estimate. On the other hand, the number of training samples that you have, the population size, is usually a lot smaller. Let's say 1,000 individuals. So what you're trying to do is solve a set of equations where you have 35,000 unknowns, and you try to solve this with 1,000 equations. So I don't know if you had a lot of linear algebra, but it's like having 3 x, y, and z variables, and you try to solve them with only one equation. That is just impossible. So you basically you should be selecting the SNPs that you're going to use. Of course, if you have to do it manually, you end up using, for example, SNPs that reach genome-wide significance, the GWAS approach, which have been bad-mouthing a lot since this beginning of the talk. So that is not going to work, like pre-selecting SNPs. On the other hand, if you just try to select thousands of them because you have 1,000 training examples, and that would work, you have a higher risk of overfitting your model, meaning that it would work really well on your training set, but basically on new individuals it wouldn't really predict anything. So the apparent success results from fitting statistical noise. You would be training perfectly on your training set, but you wouldn't be able to predict any new incoming. So the art is actually balancing the two, and one of these methods, a very popular method, and this is actually not a genomic prediction method, but actually, again, a very old school method called ridge regression. I think this is early 70s, so far before there was any talk of genomic prediction. But the idea is that normally, if you do a kind of linear regression thing, you try to minimize the error of your predictions. You try to minimize the residual value, the square of your residuals. That's what any least square estimate does. If we do that, we get into trouble. So we add a second constraint to avoid overfitting, namely that we want the sum of our estimates, beta would be the estimate of each marker, that we want to sum, the squared sum of these have to be minimal as well. So what we're basically saying in human terms, for those of you who are not following the equations, I want a model that fits very well, but it has to be as simple as possible. Any complex model that tries to follow my points is out, even though it will give me a perfect fit. And in the inverse, any model that just uses 2 parameters, just because you only need 2 points to put a line on there, the fit is so bad that is not good enough. So you try to balance the two, how good your model fits versus how many markers it's used, or I should actually say the size of the marker effect. So you need a tuning parameter that balances the two. If you see, this is the parameter that measures model fit, and this is model complexity. And if you make lambda a really big number, that means you put a lot of weight on model complexity. Well, on model simplicity, I should say. And if lambda is small, you put a lot of weight on model fit. So now the question just becomes, give me the good number for lambda, and we're gone, because all the rest is automatic. It seems complicated, but there's ridge regression is in any statistical package will do ridge regression for you. As long as you give it the lambda number, you're golden. So there are various ways. One of the ways is doing cross-validation. Just try different numbers, take part of your data set out, fit a certain lambda value, and see how well it predicts on the part that you took out. And do that just many times until you find the lambda that performs really well. Sounds a lot of work, but with these computational scripts and all that, you can automate that. It's really fast. Still, it's a bit suboptimal. You also have, remember, this maximum likelihood approach that I've already mentioned during the mixed model, where we're going to look at the likelihood of our model and try to maximize that, try to find the lambda value. And EMMA is, again, a computational optimized version of that. Just briefly, there are routines that will find the optimal lambda value for you. That's the very short one. So, and at that point in time, you have a prediction method that balances model complexity versus model fit. In popularity of genomic prediction models, I think ridge regression, or ridge regression blob as it is often called, is equally as popular as G-blob. Ironically, they're actually the same, meaning any ridge regression model can be rewritten as a G-blob model and vice versa. So why make the difference then? Well, G-blob has here in ridge regression blob, we are modeling marker effects. These betas here, these are the effect for each marker. These will all be very small numbers, slightly positive, slightly negative, but if we have 35,000 markers, then we'll get a vector here with 35,000 estimates. Very small numbers. In G-blob, we're not directly fitting marker effects, we're fitting genotypic effect, individual effects. And the difference is that we're fitting a genomic relationship matrix as a covariance matrix. So I imagine it's a bit hard to see how these two are related, but imagine if you're doing a regression with ridge regression and you see the vectors of marker scores. Individuals that are similar will have similar vectors in the markers, so they will have, in this kind of matrix, there will be very high similarity scores. So here you're fitting a kind of model on individuals based on a genomic relationship matrix, based on how close individuals are, we'll get an idea of their effect, and implicitly, as you are using the markers to make this similarity matrix, you're doing implicitly exactly the same as the ridge regression method. I have not yet found a nice way to show the equivalency without going into the mathematical derivation of the two methods, but it's basically you can rewrite the one formulation, this one, perfectly as a linear mixed model where you fit this as a similarity matrix. Now this method, or it's not a method, this form, then I'll call it, is often preferred if you're interested to get genomic predictions directly, and if you want the reliability of your predictions. Because these are, so you're looking this from the individual level. As a breeder, that is usually where you're looking at. However, if you want to make future predictions, you would be interested in your marker estimates. Now you can go from one formulation to the other, so it's not that extreme, but usually if you want to use your prediction model to make future predictions, I would opt for the ridge regression approach. If you just want to have prediction models once, you'll go for the G-blob approach. This is basically saying what I've already said. RR blob is basically equivalent to G blob. Yeah, I've said most of this, and yes, I've said that. Now, I'm not going to go into all other different genomic prediction models that have started. This one is somewhat popular because it's a catchy acronym, the LASSO, or least absolute shrinkage and selection operator. And why is it interesting? Because if you look at this formulation, it nearly looks identical to ridge regression at first glance, right? Remember, ridge regression, we try to minimize the error. We try to maximize the model fit, that's basically saying the same. And we try to minimize model complexity. That was this part, and we have to find a lambda. Now, the thing is, in this Bayesian approach, it is assumed that most of these betas, these estimates, are zero. So you take an assumption, which you don't do with ridge regression, but in ridge regression you assume that your marker effects follow a normal distribution. Here you assume that most of your markers have no effect whatsoever and are put to zero. So it's a kind of a selection, marker selection approach, but you make, of course, this assumption on their distributional form. And this is what where these models differ. I don't know if you ever heard of these Bayes A versus Bayes B, Bayes C. These are very intriguing conversations you can have with people that are doing genomic prediction on which method is best. The thing to remember is that they all differ in the assumption of how the distribution of your marker effects look like. Ridge regression assumes Gaussian distribution. Lasso assumes a very spiked distribution, meaning that most of them are zero, so a big spike around zero, and then the rest is again Gaussian distribution. And then you have lots of different assumptions, or even where they use mixtures of distribution. It can get all very complicated and so on. And in some cases, this actually outperforms. If your genetic model actually uses this distribution, it turns out you get a better prediction accuracy than using the Gaussian assumption. So there are many options to choose from. And add to the mix, which is very popular now, is machine learning methods, where one of the most easiest to understand is random forest, but going to neural networks, support vector machine reproducing kernel Hilbert space regression, and so on. These are just one of several machine learning techniques that are used as genomic prediction methods. But for all of these methods, what is usually seen that there is no golden standard. There is not one method that will win in every case. And if you want to use machine learning methods, it very much depends on the size of your training set. The smaller the training set, the more difficult it will be to find the right parameters that are needed for these machine learning techniques. So results differ. Now, this is again a study on different data sets, and it's interesting to see, because it's a bit of a contradiction what I just said on previous slide. These are different methods. Remember, ridge regression blob is like the standard method. And if you're green, you win. That's the game. If you're red, you're the loser method. So you see ridge regression blob, the most standard push the button method, wins once. And we have the most complex method, reproducing kernel Hilbert space regression. Not very catchy, but a machine learning technique wins almost every time. So it would look like, well, there's an easy choice, right? Why would we even bother doing these basic things if this nearly always wins? Well, that's the intriguing part of this paper, actually. If you look at the one next to it, it's called SVM, stands for Support Vector Machine Regression. And those two methods here, while different acronyms, are mathematically almost equivalent. So it's very strange. In fact, it's nearly impossible that this one would be so good and this one would be so bad. Now, why is that? How can you explain that? Well, both approaches need a lot of fine-tuning. Remember, like what I said with the ridge regression, that we needed to find the lambda parameter. Then we had only one parameter, but then we found a solution to find it automatically. These kind of methods need several parameters, which need to be optimized, which you need to do cross-validation. So it's a lot of work to get a good prediction accuracy like that. A lot of manual tuning and things like that. But then you can outperform the basic method. But apparently, the person who was doing the prediction was not a big support vector machine fanatic. So he didn't put a lot of effort, I think, this is just a guess here, he didn't put a lot of effort here on tuning the parameters for support vector machine. While these two methods are equivalent, you see these differences are huge. So the message here is, if you want really top-notch, you want the best prediction accuracy, okay, use one of these methods, but be ready for lots of time spent on fine-tuning. This is not an automated process. While you will spend lots of time fine-tuning, trying different combinations of parameters, if you want something to work out of the box, go for the standard methods. These will work as long as your data is not too bad, not too skewed. This will work every time. And prediction accuracy are not that low compared to the rest. That is kind of the message I'm trying to send here. And also, except if you have lots of time, like I said earlier, it seems like in literature there's a lot of focus on using and comparison these different methods The real advantage is in phenotypic data analysis and phenotypic data quality and training size, population size. That's where the real advantage is, and not these small percentages you will gain with using more complex and advanced methods. Just my point of view. Application of genomic selection. Like I said, I think I've mentioned this in my first talk this morning. Multi-trait selection of candidate varieties, selection of seedling stage, early screening, all of that. In silico generation, prediction of offspring performance, ideotype creation. So I've mentioned all of that this morning. Last slide. This is how do you incorporate genomic prediction into your breeding program. Very sorry, I have no example on forage breeding programs, but I guess this is one of the goals of the UCLEC project, to find out how to fit that in. So this is a conventional line breeding program, very much simplified. So this is like for wheat or something. You make an initial cross, you use inbreeding, try isolating lines, do small-scale trials, large-scale trials, and eventually get a variety generally assumed to take you 10 to 12 years. Traditional phenotyping line breeding. If there are people with experience on line breeding, please correct me if that is, if currently the release speed is much higher, I don't know. This is one example of how you could, this is again the same line breeding, and I included directly doubled haploids in it, because genomic selection and doubled haploids, that's like the combination of technologies that allow you to have the real advantage of genomic selection. I know very well that doubled haploids are not available in every species and in every line, but if it is available, it is a machinery that is unbeatable in terms of rate of genetic progress. So we start out the same, make crosses, use doubled haploids, so no consecutive, this has nothing to do with genomic selection. Genotype at low density, use imputation to go to high density, and then predict values using this prediction model. Do a small-scale trial, do large-scale trial, a cell or release. Of course, the left part of the model has now been added, the left part of the cycle has been added here. We need to make a prediction model. So we will need data from other efforts. That's generalized term for saying you need to do these training panel construction, phenotype them, remember balance good with bad, and so on. So a lot of effort is in here. You can also use a bit of this data to train your model, but of course not too much, because that would skew the balance. Genotype at high density, your elite material, to learn your imputation algorithm, and then you can use these models to go on. So it's a lot faster. You kind of have the time for development. You only need 6 years. These are rough estimates. But I'm hoping that you see that there's a considerable more effort needed to go in that. In effort, I mean the complexity of the operations that you have to do. So it's not something, genomic prediction, if you want to introduce it in your breeding program, that you can just do it on the side. Like we're going to keep our traditional breeding program, and on the side experiment a little with genomic prediction. In my point of view, it's all or nothing. If you have to do these additional efforts, you have to do them seriously to get good prediction accuracies. So you will have to go the extra mile to do the extra steps, and then at that point, it will allow you to increase your rate of genetic progress. This is a similar setup. I'm not going to go too much into detail. Similar setup for conventional hybrid breeding, probably not too interested, but you go again from 10 to 12 years to a similar setup where you can do it in 8 years. And I'd like to conclude again with this slide I showed you during the presentation of this morning. You see that there is quite an additional effort needed to implement genomic selection. No doubt that it will bring you an increased rate of genetic progress, but keep in mind that traditional breeding value estimation is not just a step towards, it's a crucial part. Phenotypic analysis is a crucial part of introducing genomic prediction. So with that, I'd like to conclude. I don't know how I am, quite timing. Okay.
Thank you, Steven. very, very hard day for you. Okay, I have one proposition for you to merge this discussion with Steven, because probably there will be a lot of questions, with general discussion for the end of this day. Stephanie, would you like to join us for general discussion? Yeah. So please, are there any questions for both of lecturers today? Yeah. Is that needed?
Thank you, Stephen. I'm really impressed. I like your idea to get a single phenotypic value, but what do you mean? Is it a single phenotypic value for one trait, for example, yield, or is it a single phenotypic value calculated as an index from many traits, such as yield, quality, resistance to any disease, and so on?
In the context of my talk, I was actually referring to one value per individual per trait. Sorry, I didn't make that clear, but the point is that all of these methods that I was scrolling to, if you want to go into this, they all assume that you just have one single phenotypic response, one value for one trait to do your prediction model. Well, any breeder knows that you don't have one phenotypic value, but you have multiple in multiple trials and things like that. So that's what I was referring to. Now, your question does bring an interesting topic, is then how are you going to use these genomic predictions, because this is just one trait I then train, but then indeed you'll have to combine them in some kind of selection index. And again, there are multiple theories, there are even multivariate genomic prediction models that will do these predictions on multiple traits on the same time. That's very advanced application, so this is a non-trivial thing, but that exists to do that directly. So I was referring to a single trait, but combining them afterwards is equally important, obviously. Tom has a...
I'm not sure that you are the right person to answer, but at least from a computational point of view, completeness in the genotyping data. In species with huge levels of genetic diversity, it's possible that it's just a pure biological reason that you don't find a marker. It's just this piece of DNA is just not there, right? Mm-hmm. So how would you advise, or how would your model sort of for imputing, because imputing assumes that the DNA is there. There might have been a technical reason for not detecting it, but if we look at genome resequencing studies, there's a huge amount of the genome that's just missing, right? And imputing would assume that this piece of DNA is actually there, but resequencing has shown that it's often just not there. So it's actually a negative observation is a true marker.
I understand the question, but I have to admit I don't have an answer, meaning that it's something that seems to be underlighted in most studies that I've ever encountered. I mostly, people that are doing this genomic prediction, as myself, assume that the marker matrix is there and is a given, and are not too much concerned about the biological background, which is a deficiency, I guess, and a lack of knowledge of I'll talk about myself, of my side. So undoubtedly, if you were, for example, be able to just fit the missing data, for example, as a marker itself, that might contribute something. The only thing is that you often will see that each marker has such a small effect that probably this effort that you're doing of trying to fit these missings as separate markers, like you say, I'm not sure that it would be much beneficial, but to be honest, I don't know. I've never done it, and I've never seen a study doing it. But maybe if I would Google it or Web of Science, it might be there, but I don't know.
Well, I think that the missingness is just a manifestation of the genetic diversity. Mm-hmm. It's showing itself as missingness.
Yes, so, and therefore it's informative, yes.
Absolutely. So I just would like to pass that message on. And maybe it's better just to fall back on the complete part of the matrix and forget about the empty spaces, or find a better way to
Yeah.
to give value, or
The problem is with all of these methods that you cannot, as soon as there's one point missing in the entire matrix, either the individual goes or the marker goes. But so there's no way to, and then usually you would end up with a really small training set and set of markers. So I'm guessing just ignoring every missing data point would be dramatic for your training population size.
yes.
Another question? So I have one question for Stephanie. Yeah. Our practical builders are always look for accessions, and we are opening a Eurisco database, and we are looking for phenotypic data. So when you open this engine, there are a lot of traits, same traits with different names. So plant height in centimeters, plant height with marks, so it's working. So is there possibility to rearrange that to give some template for us uploading the data to simplify the search of the data?
The problem is that such templates is not possible to made by us. So for the Passback data, there was a decision made by people who have the knowledge, and there was long discussion to came on the standard. So this have to be done by people who are working with the data. In general, what is possible in general, and what we could imagine if we get some funding or if we find the time to extend Eurisco, is to put on such different traits ontologies. So like we did it in Eurisco, that makes them more effort in performance as well, because we have to map different trait names on the same method. But in general, this would be possible and is feasible. But yeah, the thing is, with increasing of data and the standards, we will get more and more trouble in the future. So I think there's a lot of work and it's ongoing, but there's no solution in all. So not only in Eurisco, in general, at the moment.
Thank you. Are there more questions? So this is our final discussion, yeah?
So, Stephen, I have a question on the use of the genomic prediction equation for several generations. It seems that this is a difficulty because we are going to lose very quickly the prediction due to the recombination. But often when we make model to say we will have a good prediction with genomic selection, good gain. We use that for 2 cycles, or what is your advice on that?
Well, my advice is if you're going to use genomic prediction on a routine basis, is that it's a yearly process, like shown here. It's a yearly cycle of training data and making new genomic prediction models. And I know there have been studies on that try to find out how could we skip a year just to save costs and all that. But I guess that's all very trial and error, because you would have to do these kind of studies to know how much accuracy you're dropping, like the one I've shown you with the FST going. So it's a bit difficult to say, but the companies that are routinely using genomic prediction, they train every year or every season. So I'm imagining it that is to keep their prediction accuracy on a certain level.
Just a follow-up, if they do that every year and every generation, don't they lose the whole point of it to save effort and money and time? Or
Well, the initial idea was to save money and time. What in fact it turns out now is that it's a reallocation of money, effort, and time. So with an increased efficiency in selection intensity. But I've not seen a reduction. I've never seen an empty field where we used to have a trial there, but it's no longer necessary because we're doing genomic selection. No, so it's just a reallocation.
But this is only for a part of the population, so you can have a larger intensity selection. And so that's maybe where the gain is.
Exactly. The gain is having a better idea of a better selection intensity and a better efficiency. And basically, if you remember the rate of genetic progress as shown in the initial, just have a steeper slope, improving your population more rapidly with the same amount of money and effort you're putting into that. So being more efficient in general.
Another question?
Stephen, I have understood, so there are many models, and so finally, so what do you recommend? To test all these models, and or finally to choose one that we understand, we are confident we can use, and we we know how to tune the parameters, and then
I would actually vote for using one of the more basic approaches like ridge regression blob or its equivalent G-blob, because that's the standard method. If it doesn't work there, and none of the other methods will give you anything. And if you really insist, you can put effort in testing with the other more advanced methods, but that depends on how much effort you're willing to do for this little bit of gain in prediction accuracy. Like I said, in my experience, a lot more gain can be obtained by optimizing your phenotypic data collection, so trial designs and such, and your phenotypic analysis. But often there's a kind of a misconception in a lot of companies that they think that the advancedness of the genomic prediction method will make up for the less optimal phenotypic data, which is never the case in my experience. So I would go for optimizing phenotypes first. Therefore, I said the breeding value estimation approach remains very important, and then add on a standard method. And then if you still have time and effort and you're willing to do the effort, you can experiment with more advanced methods. That's kind of my point of view, but there's a lot of different views on that. So.
Okay, I am also interested with your idea to include in the training set both good and bad genotypes. No,
it's not my idea.
Oh,
it's,
no. You are reporting that it's...
I reported, yes.
Okay, and but we are speaking about quantitative traits, so do you mean that we need a sort of equal distribution of traits, or a distribution with two peaks? I mean...
No. I mean a continuous distribution. I don't know if I have one in this presentation, but you know the Gaussian distribution. Mm-hmm. And normally, in a lot of breeding programs, they only continue those guys that have a good performance. Mm-hmm. And they even don't harvest the bad ones, or even I've even been to companies where they spray them to death because, yeah, it's never going to work out anyway. So we only put effort in harvesting and drying and those that have good potential. Now, if you're making a training panel for genomic prediction, you can't do that. And even going one step further, you have to make initial crosses between parents or populations for which you know that it's very unlikely that they will ever produce something. But you need an equal representation over this Gaussian distribution. And I'm talking now, imagining again for one trait, but you have to look at all traits and get a kind of an equal distribution over their potential, over the economically important traits. And it's not an easy exercise, and it's a difficult thing to train breeders on the field to also correctly and accurately score individuals for which you know there will be bad. So use your entire scale of scoring if you're using a visual scoring tip.
So do you mean we make a training population with a Gaussian distribution, or do you mean we have equal number of each class of values?
Very good question, because that is one of the main questions they are trying to answer in the different publications that I've shown around training panel construction. And the current, it's not a consensus, but the latest thing, the latest conclusion is that what works best is having equal amounts of the tails. They call it tail selection. You select the very bad ones and the very good ones. And it resembles a bit a classic QTL study, where you would also get a parent that is resistant versus one is non-resistant, but then the quantitative equivalent would be taking the real bad guys and the real good guys. Because otherwise, if you take a real Gaussian distribution and sample from that, you'll get a lot of guys in the middle, because that's what the Gaussian distribution does. And those guys don't really contribute a lot to the genomic prediction. So that is the current state on that research, but it's really in a topic that is in ongoing research.
Just one question. So in EUCLEG, as you know, we have also the intention, or Philippe has the intention, to incorporate GWAS information into genomic selection or genomic prediction models. What do you think of that idea? Is it something that is done, and you have experience with that?
Yes, it's definitely done if you have a trait that has some very important QTLs on it that have not been fixed in your breeding population, obviously, for some reason. And in those cases where you have these important QTLs, it is actually even necessary to pre-correct for the effect of these QTLs so that they do not dominate your quantitative part of the model. So yes, it is being done. Yes, I have experience, but I have to admit that it's rare for most quantitative traits, because the large QTLs have generally already been fixed by traditional breeding. So I rarely see these big QTLs being found in advanced breeding pools. But of course, we're not in the advanced breeding pool stage yet. So I find it an interesting idea, but I cannot promise that it will deliver something.
More questions?
Yeah, it's more a comment than a question. So even if we don't go for a genomic selection immediately, the use of molecular markers to calculate the kinship would be very useful to actually go on your red line so estimate good breeding value.
In my point of view, yes. And actually, at that point, one could argue that you're already doing genomic prediction. It's very close to one another. There's no clear separation. Yeah. But in my point of view, it would be an interesting step to take.
Someone more?
It may be asking a question again that has already been asked about the good guys and the bad guys.
I'll try to reply
Is it-
consistently.
Can genomic selection help to fish out carriers of really deleterious things that you would actually like to remove from the baseline of your program, or is it still good to drag them along so that at least you can recognize them at some point? Do you know what I mean?
Yes, and it's also very related to the topic of retraining.
Yes.
Because normally you would automatically be doing that by selecting your top candidates, and at that point the bar of what is bad also changes, because if you don't change that, your training population is no more reflecting your breeding population. So you're shifting the bar each year normally by just genetic progress, and then what used to be bad a few years ago is now way off the scale, and what used to be good is now maybe bad, maybe an extreme situation. So you'd be doing that implicitly, and by retraining each year, you would have a model that is more adapted to the new situation where they're. So I'm guessing you're doing it. The only way, moments where I actually see negative selection going on with genomic prediction is when the prediction accuracies are too low to do positive selection, where you go under accuracies of 0.4 and so on, where it's too dangerous to use it as a positive selection tool. But if you look at it from the negative point of view and you remove like a limited percentage, then that's acceptable. But I haven't actually seen using genomic prediction to really do with the goal of negative, but you do it implicitly.
So it was quite a long day, especially for Stephen. If there are no more questions, yeah.
Thank you.
At the final end, I would like to thank all lecturers in these 2 days for tremendous effort they made. And I want to give them one huge applause again. And for all of you, I would like to thank you for participation. I wish you safe back home, and I hope we will see each other in some similar occasion. So thank you.